Hi, everyone. So my name is Granera, and I'm an intern at the Kreitzer lab. And today I will be talking to you about brain circuits involved in OCD. So first off, obsessive compulsive disorder is a psychiatric disorder that has three main symptoms: obsessions, compulsions, and anxiety. OCD patients have differences in several brain regions, such as the amygdala. In the Kreitzer lab, we use mice models to try to find specific brain connections that can cause OCD symptoms. In the case of mice, it manifests as compulsive grooming. So what do we know? We know that a subpopulation of cells in the medial amygdala promote grooming. We also know, based on prior experiments by my mentor Robyn, that activating the ITC amygdala might increase grooming. However, we don't know if the ITC amygdala projects to the medial amygdala, and we don't know if this projection is involved in grooming. Essentially, what we're trying to find is if there is a connection between the ITC and the medial amygdala, and if this connection controls grooming. So we hypothesize that if a projection from the ITC to the medial amygdala exists, if we increase ITC activity, we will also see an increase in grooming behavior. So in order to find out if our hypothesis might be correct, we conducted two experiments. In our first experiment, we used DREADS to simulate ITC activity, and DREADS stands for designer receptors exclusively activated by designer drug. And this experiment helped us investigate if increasing ITC activity will also lead to an increase in grooming. So the first thing that happened was that Robyn injected and the first thing that happened was that Robyn delivered the virus using during surgery and injected it into the ITC. After a few weeks of recovery, these receptors were fully expressed in the ITC. On day one of our experiment, we injected mice with a vehicle drug. This will be our no drug condition and recorded how much they groomed for 30 minutes. On day two, we injected mice with clozapine oxide. This will be our designer drug and recorded how much they groomed for 30 minutes. So as a result, we can see in the graph that on day two, under the drug condition, mice groomed about 100 seconds more than on day one, where they were only administered the vehicle drug. So activating the ITC does increase grooming. However, future experiments are needed using a more specific viral strategy. So for our second experiment, we used retrobeats to map the medial amygdala to ITC amygdala pathway. And this experiment helped us investigate if the ITC amygdala projects to the medial amygdala. So retrobeats are a retrograde tracer. And the first thing that we did was surgery, and Robyn delivered the beats to the medial amygdala through a glass pipette during surgery. And after a few weeks of recovery, the tracer had enough time to travel to its projection region, and the mouse was ready for brain retrieval. After brain retrieval, I sliced the brain and mounted the slices, and later we inspected them under the microscope. If our hypothesis is correct, then we would expect to see beats in the ITC amygdala that traveled from the medial amygdala. So when we inspected our brain slices under the microscope, as we can see, we can notice some fluorescent spots. This one right here corresponds to the injection site in the medial amygdala, and this one right here corresponds to retrobeats that we found in the ITC dorsal cluster. We were expecting it to see in the ventral cluster, but we only saw it on the ITC dorsal cluster. And this right here is a close-up of that. So this finding suggests that the ITC does project to the medial amygdala. However, further testing is needed. So when we increase ITC activity or map a projection from there, we are not only stimulating the pathway to the medial amygdala, we are also stimulating cell activity in other pathways to regions that lead to the BLA or the central amygdala. So the next step would be to use a more specific viral strategy that targets exclusively the pathway between the ITC and the medial amygdala. And thank you so much for this opportunity. And I want to thank the Kreitzer Lab, my mentor Robyn, and everybody who helped to make this possible.
Great job, Jenny. Thank you. Any questions for Jenny? Oh, there's a quick question. Can you talk a little bit more about the specific viral strategy you used?
Yes, so in this case, we use DREADS, and Robyn plans to use optogenetics to repeat the experiment. So we will use mice with channel rhodopsin expressed in the ITC, and then we would implant a light fiber in the medial amygdala and repeat this experiment and to see how much mice groomed with this viral strategy.
Wonderful.
Thank you.
Thank you, Jenny.